All right, we're live.
Okay, good morning. Thank everyone here, and we've got over 30 people now on board. So thank you for taking the time out this morning, and really thank you for getting involved by volunteering for the BPC Citizen Science program. We're really excited about the program this year. Last year is a calendar year that I think we're all happy to have turned, and it did slow down a little bit of our testing, but we're still able to test 13 ponds last year with APC, our partners' helps, of course. So we're excited about that. This will be our 5th year of testing and sampling ponds in the town of Brewster. So we started in 2017 testing 4 ponds, and that testing has grown this year. Our plan is to test 18 ponds, which is, as Kevin and I talked all winter long, a pretty aggressive number, but we identified ponds that we really thought were important. The number happened to be 18. And then it was, okay, we're going to have to get many volunteers to do that. So the growth, the message that we're getting out to you guys has been heard. So thank you. And the real truth of it is, if you don't do the work, it doesn't get done. There are not enough scientists or people in Department of Natural Resources in any town in Barnstable County to do the work. So that's why citizen science, really is important. So I think you know that, but I really like to say it out loud. The other thing that we found to be important that doesn't always happen is good communication. So after you're doing all your work sampling, Kevin and us are doing testing and analysis, it's important to communicate the results. So it's like if a tree falls in the forest and no one hears it, did it happen? So too often maybe we're gathering results but not communicating them as well as we could or should. So we're learning by that. And so we're going to communicate immediately out. You guys are going to be the first to know. The volunteers will hear the information and get the analysis results from me via APCC within the week following our testing. Then I'll send it out to our BPC membership, which is over 800. We'll also send out to BPC contacts that we know at the various ponds in town. So 18 ponds tested, and we have a number of pond association representatives that we'll communicate to, with the hope that they'll then tell their neighbors what we found out. So that's the only way any of this works, is really also to have good communication. And then last but certainly not least is we will communicate the results to the Brewster Department of Natural Resources, Chris Miller and his group, and to Amy Van Hoen at the Brewster Department of Health. In the event that any pond advisory, if we're suggesting a pond advisory due to guide value cyanobacteria levels that are higher than Commonwealth, we will communicate that, of course, to Amy Van Hoen and the Department of Health. It is their responsibility, and they're accountable for the posting. But so we're here as sort of a sounding board. So that's kind of, my thoughts there. The agenda this morning, we hope to keep it to about 60 minutes. I'll turn it over to Kevin. He'll talk about the science of cyanobacteria, and then I'll review the scheduling. And the scheduling has been interesting and a little complicated, in traffic control around Gwen's garage. At the end of my piece there, we're going to show a sample video, not a sample, we're going to show a video of the sampling process, which will sort of correspond to my talking about the process. And again, the whole thing is being recorded, so you'll be able to see it as often as you want, particularly the video pieces of it and the science. So anyways, I'll turn it over to you, Kevin, to talk about the science of cyanobacteria.
Awesome. Thanks, Marty. And I just want to thank again all of you for coming. Seeing a lot of familiar faces out in the crowd. It was great to work so closely with all of you back in 2019, back when we were in the Brewster Natural Resources Lab, and going out with Roy to sample Upper Mill and Walkers. Always just a pleasure to work with your crew. And I believe we're going on our 5th year now of cyanobacteria monitoring with our partnership with BPC. And there's been a tremendous amount of data, tremendous amount of research, awareness that has come from that excellent relationship. And we've documented a number of blooms, been proactive about identifying blooms and transferring that data and information onto you guys and Amy Von Hohn and the DNR and others in the community to be aware and take action, identify which ponds are having troubles throughout the season. And it's really a tremendous program. And as Marty said, it can't be done without all the help from all you folks, especially in a season where we're really looking to expand to a magnitude that we've never gone to before. So we're really happy to have you all back and reignite the partnership of your citizen scientists' abilities to help us reach all the ponds that we want to reach. So yeah, as Marty said, I'll start off with talking about the science of cyanobacteria, and then we'll go more into the program and the protocols for collecting samples and yeah, take it from there. So just as a, it might be a refresher to some of you, but cyanobacteria are common aquatic microorganisms. They're in the phytoplankton community of not just ponds, but in marine areas and everywhere around the world where there's water. You might also hear them called blue-green algae or blue-greens. Those are in reference to cyanobacteria. They're a very old group among the first photosynthetic organisms on Earth. We have them to thank in part for our oxygen-rich atmosphere. They're a very diverse group too. There's thousands of genera of cyanobacteria, some like spirulina that humans use as health supplements. But what we're looking at are the genera involved in our lakes and ponds. And for this region, there's about 10 or so common genera that we see in and out of that, probably around 3 or 4 that we see pretty commonly. And they are very widespread, very common. A common misconception about our program is that we're testing to see whether or not there's cyanobacteria or not in a ponds, see if it tests or fails for the presence of cyanobacteria, but cyanobacteria are always there, in the ponds that we monitor, and in normal conditions, quite harmless. The only concern is when the population spirals out of control and they grow to dominate the ponds, the phytoplankton, everything gets out of balance, and that's where the concern comes in. But for most of the year, for most ponds, they should present no issue for humans. Some of them are nitrogen fixing. You can see in the top right corner, there's 2 genera of cyanobacteria, 2 of our more common ones. The one that looks like a beaded necklace, you can see some white beads and then some bigger green ovular beads. Those are Dolichospermum. They're known nitrogen fixers. Then the one that's a community of small dots, that's Microcystis, which produces different toxin concentrations than Dolichospermum and behaves differently than Dolichospermum. Some genera have buoyancy control, that they can regulate where they are in the water column using gas vesicles, so they can migrate to the surface when they need to photosynthesize and then regulate down to the bottom. There's also benthic cyanobacteria that more specifically proliferates and lives more on the bottom depths of the ponds. And as I said, the big concern is when they form blooms, whether it's dense surface scums or they turn the ponds bright green, cause the visibility of the pond to really decrease. These are all concerns, as when they get to this point and they're really taking over the ponds, they can produce toxins called cyanotoxins that can be harmful to humans, pets, and wildlife. I'll talk more about maybe the human impacts on the next screen. but they can also have effects throughout the food web. As your group has been very involved with Nancy Leland with her trophic status studies and understanding the effects that cyanobacteria can have on the rest of the food chain, as cyanotoxins can accumulate in fish tissues like alewives and in zooplankton that graze on them. And it's very interesting to see the effects that cyanobacteria have, not just locally in their ponds, but also throughout the rest of the food chain. So yeah, why are we doing this all? For the public health impact, it is the toxins that they create is just very important to know and understand. There are multiple methods of potentially being impacted by cyanobacteria exposure. The most common is through consumption of pond water that might include cyanotoxins, including microcystin, which is a hepatotoxin. That means it can potentially damage the liver and other internal organs. There's ways of coming into contact with it through just skin contact. It can cause rashes. There's studies being done by Jim Haney and Nancy Leland and you folks on aerosolized cyanobacteria and bioaccumulation. But largely the main thing that we're worried about as far as human or pets interacting with ponds is incidental consumption of pond water containing toxins created by cyanobacteria, as these cause the most immediate effects, the most concerning effects, and they can happen through a pet drinking water at the shore while they're on a walk, incidentally consuming some of that pond water, or going swimming and grooming themselves after. Humans, when they're swimming, can incidentally consume some pond water. Children and people with certain conditions can be more at risk for cyanobacteria exposure. So we always want to be proactive in taking this all seriously when there's blooms occurring, to try to limit your contact with your pond during that time that it's occurring. There's other impacts that cyanobacteria blooms can have, largely just degrading the pond as a whole and as a habitat. It can block light and make the pond very turbid, visibility to decrease. That isn't good for any of the anything that needs sunlight to grow, any plant that might be growing at the bottom. It also lowers the oxygen, especially at the lower levels of the pond, and that can potentially cause fish kills for anything that swims and lives in the benthic areas of ponds. And yeah, all of this degrading habitat for everything interacting and living in that ecosystem. As we know, some of the causes are largely nutrient accumulation coming into these ponds over years, whether it's septic influences or excess fertilizer use, or improper buffers between your property and the ponds, and just accumulation over time of nutrients and organic matter. Temperature also plays a role. Warming temperatures favor cyanobacteria. Certain weather conditions can kick things off. So as climate change progresses, this is something that we want to continue to keep an eye on and continue to monitor and understand. So as an organization, for us, in working with groups like you all, is to fill in these gaps for monitoring. Without us, for many towns, potentially this work might not get done in terms of monitoring each pond and their concentrations for cyanobacteria, and being proactive about when blooms are forming and making sure the right people know that when the blooms have formed, to promote recreational safety, to keep people safe, let them know when to maybe stay away from their ponds for a week or two, and then come back once conditions are looking all right again. This can help also for resource managers identify which ponds might need some help for rehabilitation or just better conservation and protection. Cyanobacteria proliferation and impairment can be a sign of eutrophication and just general degradation of a pond's health. So when continuous serious cyanobacteria blooms are occurring, that can point to a need for resource managers to take another look at the ponds. And we want to continue to educate and spread awareness of cyanobacteria as kind of how to be safe with it, but also just know that when these blooms form, and we're putting our data out there about maybe when advisories should be posted. If your pond has a seasonal cyanobacteria bloom, we want to help you learn how to live with that and understand it. It's not a reason to stay away from your pond, and change your behavior with the pond, for the future. Largely, if this happens, on a one-time basis, you might each summer you might have to stay away from your pond for maybe 2 weeks, and then the rest of the season it could be perfectly fine for your normal interaction. So we want to spread awareness and understanding, and make sure people are aware of to take this seriously, what to look for, but also to be aware of just the different behaviors that are good for interacting with the pond that might be having cyanobacteria blooms. And we're looking to expand this year to 125 ponds, potentially more in the 2021 season, including our 18 in Brewster. We've been documenting the accumulation of blooms and toxins produced by cyanobacteria in each of these ponds going back from when we started in 2017 to 2018. And the data that we collect is all data that we pass on to groups like you all and to town health officials and resource managers so that they can take action when necessary to post advisories for that pond. Some of the information that we're passing on is they can compare directly to the Massachusetts Department of Public Health standards for cyanobacteria. So they can all be aware of when problems are coming, when these problems are happening, and they can feel good about taking action based on the data that we're collecting to be proactive and efficient in their means of posting advisories and ensuring that there's awareness and understanding of what's happening in each pond. So our protocol that we've been using and adapting and improving over the years was first developed between the EPA's Cyanobacteria Monitoring Collaborative and our great partners at UNH, Nancy and Jim and their whole crew. It involves first taking field notes and scanning for cyanobacteria scum, and then taking your samples. Marty will go into all this kind of sampling procedure in the next section, and then we'll also have a training video. This just to give you an overview of the whole thing. So after you take your field notes, you take your two types of samples, a net tow, as you can see here in the picture on the right. This is collecting bloom forming colonies of cyanobacteria, or BFC. These are the fairly large colonies of cyanobacteria that we can track over time and predict when they might accumulate to a point of creating bloom conditions. The other one that we collect is a unconcentrated sample taken through a tube called whole lake water, or WLW. With this, we can see cyanobacteria just in a standard unconcentrated sample. We can also take that sample and filter it through 50 micron mesh to then see how much of the very small cyanobacteria there is, or picocyanobacteria. So there's a lot of great information that we can collect from just these couple samples, and looking at the very large, the very small, and everything in the middle. And then if there's a cyanobacteria scum present, we also like to have that sample too, so we can analyze it and ensure it is a cyanobacteria scum or bloom accumulation. It may be not. a pollen scum, as you might be seeing on your ponds last week, this week, and it's really strong. We then take these samples back to the lab and perform a microscope analysis on them all to identify composition and dominance of our cyanobacteria. This is so out of the genera that we look for, we identify which genus might be, really the dominant genus in the sample. Say there's 90% of Dolichospermum. It gives us a good idea of kind of maybe what toxins are being produced, because each genus of cyanobacteria can produce toxins differently, and different toxins as well. So it gives us a good idea of what to expect, especially in tracking that over time and how the population is changing. We then freeze our samples to expose cells to perform a pigment analysis on them through fluorometry. This can give us the concentrations of cyanobacteria in the sample, so we can know who's there through the microscopy, which genus of cyanobacteria we're really seeing in the sample, and how much of it is there. And bringing this together and tracking that over time can give us a lot of information, like I said, in terms of toxins to expect, population dynamic changes, and when to be concerned, in those times when perhaps a bloom may be forming. All of this data gets entered onto our server. We have regressions that can estimate toxicity. When available, we'll also perform tests for the microcystin toxin separately to ground truth to compare with what we're seeing in the regressions. And then UNH might also, after the season, do some additional toxin analysis as they might be interested. But there's a tremendous amount that we can learn, and the data that we collect here to really understand what's happening in each pond at any given time. And it's really a fantastic method. So at this point, I'll turn it back to Marty to go over the sampling protocol.
Thanks, Kevin. For lay people, since we all don't have microscopes and equipment necessary to do analysis, I was just thinking when Kevin was talking that I use kind of 3 simple rules when looking at a pond, and you can talk about this and talk to your neighbors about this. And one rule is if the water is green or greenish or covered with scum, avoid it, please. Keep your pets, particularly small pets, but pets in general and children, out of the water if you see that the water is greenish or covered with scum. That's kind of rule 2. And then 3, sort of to do a turbidity check when you don't have a turbidity tube or a Secchi disc handy in your back pocket, I say wade into the pond to about your knees, give or take, mid-thigh to knees. And if you can see your feet, fine. If you cannot, then it's very fair to say avoid the water. So that's sort of a turbidity check. And then report that to me, and I'll get that to Kevin, and we'll take a look. So just kind of 3 simple checks. Since none of us have equipment in our back pocket, but okay. So here is the first picture of what I call the sampling process, sort of the nuts and bolts of everything. And you don't need to have a copy of this, though I've met with 6 or 7 different pond groups this past week who've not done the sampling yet and got around with them and handed out papers. You don't need that because the sampling process will be, I put it in each of the clipboards and encased in plastic so it doesn't get wet. So that'll be there. And when you're collecting your kit, you're going to be able to have this exact PowerPoint here with you when you go out. So it'll be sort of your operational guide, like step 1, step 2. So Kevin, next screen. So here's step 1. Pick up the kit at Gwen's garage. Gwen's on Route 137, Long Pond Road. So her address is 1247. She's a house or so after Freeman's on the left going towards Harwich. So what we have at BPC is 3 complete cyanobacteria sampling kits. And our plan, as I said earlier, is to test 18 ponds. We'll be sampling the Brewster Ponds on Monday, Tuesday, and Wednesday every other week, beginning this coming Monday, the 24th. So 6 ponds are tested on Monday, 6 on Tuesday, 6 on Wednesday with 3 kits. And in order to get all that done, we have an 8 a.m. shift on Monday, Tuesday, and Wednesday. 3 teams go out, return, and then a 10 o'clock shift Monday, Tuesday, and Wednesday. So 6 ponds a day times 3 days, and we're able to accommodate, we're able to accomplish what we want to accomplish. The key really is sort of the traffic in and around Gwen's garage, which is basically, the nuts and bolts. But if you're an 8 a.m. shift, please be at the garage by 7:45 so you can pick up the equipment. Whoever on your team is going to the garage, pick it up at 7:45 so you're able to drive back to your pond by 8 o'clock and meet the other team people who will be at the pond. And then the 8 a.m. shift really needs to return the equipment by 9:30 at the latest, because you're going to get back to the garage, you'll clean the equipment, hang it back up, and then the 10 o'clock team will come in at 9:45. So it is a bit of a traffic thing, but quite honestly, that hour and a half beginning when you start testing at the pond at 8 and get back to the garage at 9:30 is going to be an easy accomplishment. After you've done this 2 or 3 times, it'll go very quickly. So I'm not really worried about the time, but those are important times. Put the kits exactly back as you found them in the garage. So you'll see when you go into the garage, there'll be columns of kit 1, kit 2, and kit 3. Grab whichever one's available. Some other team may have beaten you, so it doesn't matter which kit you take, just take a complete kit out and go out to your pond. So the kit does include all of these. I don't necessarily need to read these to you, but make sure your kit that you're taking out does include all of this. Again, the clipboard will have this PowerPoint right on it, encased in plastic. You'll have a pen and pencil. There'll be ice packs at the bottom of the cooler. This is important. Please label the 3 bottles you'll be taking before you leave the garage. You don't want to go out there with bottles that are not labeled, and you're not going to know which is which when you put the samples in. So 1 brown sampling bottle, they're all 500 milliliter bottles, but 1 will be labeled net. 1 bottle will be labeled grab or whole lake water. And the third will be a scum bottle, the very bottom bullet I have on this slide. I don't expect you're going to be collecting scum in the beginning. Hopefully we won't be, but if you see a real thick greenish scum, just scoop up the scum in that 3rd bottle labeled scum. Okay. There will be a hold on back, just go back. Yeah, so there will also be a water thermometer in the kit, a plankton tow net in the kit, a turbidity tube, which looks like a periscope tube. At the bottom of that tube, since we're all sampling, except for 2 ponds, all of us are sampling on the shoreline. Turbidity tube will have a little mini Secchi disc at the bottom of the tube. You can see the tube is the bottom picture on the left. There'll be a clear plastic tube for the grab and the scum bottle. Okay, next. So that's the contents of the kit. The process is really simple, but it does require detail and some attention to that detail. And what we suggest is you guys have 1 person on the shore, or you can be weighted in the water, but 1 person with the clipboard and the data sheet. And Kevin, if you could go to the next slide. Yes, here's a picture of the data sheet, which many of you have already seen. If you haven't yet, you'll see it on the clipboard along with this PowerPoint. So you'll fill out the data sheet. I would suggest strongly that you bring a smartphone. Everyone has phones, so bring your phone with you. You'll be able to get air temperature with your phone. You'll be able to get the actual GPS coordinates of the site at which you're collecting from the phone. So I won't go through every element here, but really the important elements are that the top is just identifying the water body. So Baker's Pond with the coordinates, May 26th, 10 a.m. shift, present Marty Birkin, Cameron Ferguson, whomever's present from your team. Then there's a weather section, a water section, and a biology section. Circle each of your responses, your team responses in each of these 3 sections. Some of them are a little bit subjective, like the wind speed. there'll be a wind speed definition page that you guys will be able to take a look at and sort of just make it a majority vote on what you think the wind speed is. Is it calm or is it gusty, et cetera. The wind direction will be either, you don't have to worry about north or southeast or northwest, just onshore or offshore direction. When it rained last, current rain intensity, sky conditions. Again, some of these are subjective, so just make it a kind of a team vote. Cloud coverage, the same thing. Water is the next section. Again, wave intensity, water temperature. Here's where you, I like to throw the thermometer into the pond right at the very beginning when you get there. Just make sure it doesn't float away on you, but throw the thermometer in the pond and just let it sit. The longer it sits, the more, I think, credible the temperature is. Everything will be Fahrenheit, whether it's air temp, water temp. And then if you're smelling something, strength of the smell, turbidity of the water. Since all of us here, except for 2 ponds, will be testing from the shore, you'll have that turbidity tube. And the tube measures 1.2 meters in depth, which is roughly 3.5 feet. You fill the tube up. We can go back to the prior slide now, Kev.
Yep.
Please. If you can do that.
There we go.
So now the data sheet is being filled out as you go along by at least one person, but sort of a team vote on the subjective. The clarity is an onshore turbidity tube. Fill it with water, let the water settle, let the little waves in the tube slow down. Take a look into the periscope down towards the Secchi disc. And when you can, if you see the disc right from the top, then you know your measurement is 1.2 meters. If you cannot see the Secchi tube, then just release a little bit of water from the clip at the bottom of the tube until you can see the Secchi clearly, and then mark down, well, this is 0.9 of a meter or 0.5 of a meter. So the clarity tube exercise is pretty simple, but important. Okay, next. So then the next thing you do, and I put this together this way because I think operationally it makes sense and it's easiest. This is the grab of the shore water that we call whole lake water. So on your already pre-labeled millimeter, 500 millimeter bottle, you'll have grab whole lake. You'll put your pond, Baker's Pond, the date May 26th, on the masking tape. Masking tape and markers are all in the garage, like I said. Get that done before you leave with your equipment. Then when you're at the pond, for all of the equipment here, and I didn't say this for the Secchi tube, which I should have, rinse everything 3 times. Rinse all the equipment 3 times, rinse the bottles 3 times before putting the samples in. And that really, it cleans the equipment, but it also conditions the equipment to the specific pond that you're at, which is important. So rinsing the bottle and then the tube, and then you take a clear snake tube, which I can't show you here, but you'll see in the video. There's a clear snake tube in the kit. And what you'll do is you'll get yourself out to, at least mid-thigh or waist, and rinse that tube, but then collect water in that tube. Place it in the water into the bottle. You might have 2 people in the water, one handling the tube and one handling the bottle, until you get a little more familiar with it, and collect enough water in the bottle to fill it at least 3/4. Then put the sample in the kit right next to the ice pack, which will be at the bottom of the kit. So that really is the whole lake grab. In the past, it was a little simpler because we just took the 500 millimeter bottle and dunked it. Now we're using a snake tube, a clear snake tube, to collect that grab. Okay, put it in the bottle, put it at the bottom of your kit. Mm-hmm. All right, next is the step 4, and this is sort of the, I call it the athletic one. It's the net sample throw. Here, you'll take your second bottle, same size bottle, label, date, and pond. Rinse that bottle 3 times with the pond water, flush it out, rinse the net, dunk that whole net into the pond 3 different times with the clip at the bottom open. So when you're dunking the net and then raising it, the water is draining from the sides of the mesh, but also coming out the bottom of the tube. So you're cleaning and conditioning the net that way as well. Walk into the pond again, mid-thigh, maybe deeper if you're more comfortable. You can see the bottom picture on the left. Chuck Medansky's in, it looks like about to his waist, and he's throwing the net. When you release, throw the net 10 meters, 10 meters, that'd be a throw. 3 meters, which is 10 feet. I like to throw it horizontally because it just seems easier for me and more effective, but you'll find your own method. And then pull horizontally the net back to you, like a fisherman might be pulling in his line, making sure that the plastic cod, which is at the bottom, you can see it in the top picture, the plastic cod end is a cup which will hold water. Water above that is in the mesh and will drain out ultimately. So pull the net steadily, 1 foot per second, which is not easy to measure, but just kind of with the fluid motion, bring the 10 feet in back to you. And then you'll raise the net up, rinse the side of the net 2 or 3 times by dipping in and out of the water, and also keep the top ring out of the water. So now at the end of this, you're going to be holding up the net with the cup filled, and that cup contains about 250 milliliters of pond water. And you'll open the clip at the bottom, drain that water into your bottle. And so you'll need 2 tosses really to fill up the 500 milliliter bottle. Pretty simple, but a little, it may seem a little clumsy at first for you to do it. And then you repeat the net throw to get your second collection in the cod end. Rinse off the side of the net as well before your second toss, and then go through the same process. Bring it in, putting the sample in that brown bottle next to the other sample near the ice pack. And that's the net throw and collection, pretty much. Step 5, the last step is returning your kit and samples to Gwen's garage. So the 8 o'clock team should be really must be at her garage by 9:30. So the team at 10 coming in at 9:45 can grab everything. Leave the net and grab whole lake water bottles that are labeled. There'll be a larger cooler in the garage. We're going to collect all the samples for each of the days. So put your 2 sample bottles. If you have a scum, you'd have 3 sample bottles in that cooler in the garage. Your data sheet that's been completely filled out now, you would put in a folder I'll have on top of the desk in the garage. So put your written write-up about what you saw in that folder, and then leave the kit on the table just as you found it. So the next 10 o'clock team can come in and grab it. You'll also need to be washing all the equipment. So wash the plankton tow net, the Secchi tube, and the whole lake water tube, at Gwen's garage, right on the side of the house. There'll be an obvious place where a hose where you can wash everything down. So wash it down pretty thoroughly, take you 5 minutes, and then hang the net back up in the garage as you picked it up, and put the turbidity tube, stack it up against the wall and back of your kit, and put the snake tube for the whole lake water back in the kit. And that's it. So the kit's all built again and cleaned out for the next team coming in. If you see anything unusual, smell anything unusual, see something you're unsure of, write that in the written report, but also take a picture with your phone and then share that with me. And you guys have my email and phone number, so that's easy to get in touch with me. And I'll probably, most of these days, I'll be running around either at ponds or in the garage. So just get that information to me. Lastly is I'll just for your info, in the past, we had each team take their samples to the Brewster DNR office when we had the lab there. With 18 ponds, we don't really want 18 cars coming in and out of APCC. So at the end of each day, at the end of Monday's, Tuesday, Wednesday's test, I'll take the 12 to 15 bottles to APCC. And so it's important even though the 10 o'clock team has no one behind them, it's important for you to get back to the garage with everything done, or get back to the garage by 11:30 so that I can then include your samples with the 8 a.m. group samples and get them over to APCC prior to noon. That's pretty important. So it's, it's a little bit of clockwork, but if you just kind of follow those guidelines, I think it'll work out really well for all of us. Now, when we get to testing 30 ponds, it's going to get even more complicated, but that's pretty much step 5. That's the entire process. And Kevin, if you go to the next, we put this in here before. I think Karen Malkus Benjamin or maybe had given this to us a few years back, because we had a lot of questions about subjectivity of wind definition. So this might help you out. Next. Here is the schedule, which I sent to everyone here independent of this morning. So you all have it in your email. If you haven't seen it yet, you could grab it and print it. But it is sort of the traffic schedule, and there's 5 columns: pond name, assigned team members. So Baker's Pond, myself and Cameron Ferguson. The first person in the list I've designated, and I've talked to, I think, everyone on this list I have. The first person is sort of the team captain, as I put it. If you guys don't like the team captain designation, you can do what you will, but these are, so for anyone on the team, go to Jan McCann at Blueberry Pond, Chuck Medansky at Cliff Pond, etc. Phone numbers for each of the individuals in order. So you guys have each other's phone number. The schedule start date, and then every other week. So for Baker's Pond, we start at Wednesday, 8 a.m. and then 2 weeks down the road, I wasn't going to put every date in there. You will all be able to put that on your own calendars. So we'll be back out. The Baker's Pond team will be back out Wednesday, 2 weeks down the road at 8 a.m. again. That's critical. So the day of the week and the time that you're assigned to here carries on every other week throughout the entire summer. So not that complicated, but I wanted to say it out loud. And then the 5th column to the right is the site for sampling. And this is important too, may seem obvious, but the samples always have to be taken at the same site. You're going to have that GPS coordinate rate identification, and what you see here ultimately is the site that you'll go to every other Wednesday, Monday, or Tuesday at your time to the same site. So that's pretty simple, but important to say. We don't want you testing at different sites. So that is, I don't think there's anything else I need to point out on that schedule. So let's go. You guys can have questions at the end if you have them, but let's go to this training video, which will kind of parallel what I said in the written slides, and it's done by APCC and Rebecca Miller. Thanks, Kevin.
Yeah, just make sure to let me know if you can hear the audio here.
Since 2017, the Association to Preserve.
How's that? Can you hear it?
I can.
Increase the volume a little bit. Increase the volume.
Yep.
Since 2017, the Association to Preserve Cape Cod Cyanobacteria program has relied on citizen scientist volunteers to collect water samples across Cape Cod. The sampling process is simple and straightforward, but requires a keen attention to detail. Before we begin, thank you for your interest in assisting APCC with this critical program. Your help allows APCC to increase the number of ponds in our monitoring network. That will improve our region's understanding of the cyanobacteria trends in our beloved freshwater ponds. When setting out to collect your samples, plan to be comfortably finished before 9:30 a.m. at the latest.
And of course, for your schedule, you guys will be on your own your own kind of two paths, your 2 schedules. For other groups that just have kind of one shift, we have them finished by 9:30. So don't worry about that timing.
Thanks for saying that, Kevin.
The bloom formations are more likely to be intact in the early morning before the water is disturbed. It is also important for APCC to obtain samples early in the day so sampling process can begin as soon as possible. APCC likely has a planned meetup location for you and your group to pass off your samples, whether that is at our office in Dennis or perhaps at the Barnstable County Water Lab in Barnstable. Drop off your samples to the APCC meetup location by 10 a.m. so sample processing can begin on schedule. The sampling equipment that should be brought to each sampling event include 1 student plankton net, 1 5-gallon bucket, 3 500 milliliter sample bottles per pond sampled that day, 1 1-meter tube with a weight on the end, 1 digital thermometer, 1 clipboard with a pencil and field data sheets, 1 cooler with frozen ice packs, rubber boots or waders, rubber gloves, a digital camera or cell phone with a camera, and other personal equipment such as sunscreen, bug spray, hat, etc. Label the 500 milliliter bottles prior to heading to the pond. 1 bottle should be labeled WLW, standing for Whole Lake Water, as well as the pond name, date, and town. The second bottle should be labeled NET with the pond name, date, and town. The third bottle should be labeled as Scum Sample with the pond name, date, and town. It is important to note that if you see a visible cyanobacteria bloom or cyanobacteria scum on the surface of the pond, it has a chance of being toxic. Before you do anything, take photographs of the pond and any bloom or scum you may observe. It's good to get the photo before it is disturbed by sampling. Please wear gloves while sampling to avoid contact and be sure to wash your equipment as soon as possible after sampling. If there is a visible bloom or scum, use the third bottle to collect some of the surface scum. Again, wear gloves to protect yourself. Collect just the scum. No extra water should be added. Place the bottle into the cooler and APCC will identify it. Walk into the water to about knee to thigh deep. Be careful not to stir up the bottom. Fill the WLW bottle with water and dump it. Do this 3 times to condition the bottle to the pond you are sampling. Repeat this same procedure with each of the NET bottles. The first sample to take is the Whole Lake Water sample, WLW. Take the 1-meter tube with the weight on the end. Insert the tube into the water, making sure to avoid resting the tube end on the bottom of the pond. Place your thumb over the opening of the end of the tube that is above the water. Raise the tube out of the water, dispense that sample, and then collect another sample in the tube, repeating the process 3 times to condition the tube. When conditioned, collect a sample with the tube and then place the water end of the tube over the sample bottle and empty the water into the sample bottle by removing your thumb. Repeat this until you have filled the bottle to at least 3/4 of the way. Replace the cover on the bottle and place it into the cooler where it will remain on ice until it is processed by the lab. Time to collect the net sample. Just as you condition the NET bottle at the start, the plankton net itself must be conditioned. Do this by opening the clip at the bottom of the net so the water can flow through the net uninterrupted. Dunk the net completely into the water and let the water run all the way out of the net 3 times. When conditioned, close the clip at the bottom of the net and dunk only the bottom of the net into the water, so the net is filled with water. By doing this, a little bit of weight has been added which will ensure when you throw it, the net will land into the water the correct way. While holding the end of the rope that is connected to the net, throw the net across the surface to the water about 3 meters horizontally away. This is about the length of the rope attached to the net. When the net hits the water, begin pulling it towards you along the surface of the water at a rate of 1 foot per second. Once you have the net fully reeled in, dunk just the bottom of the net into the water to wash away any cyanobacteria that may be clinging to the sides of the net or the bottom of the net. Be sure not to reintroduce any new water into the top of the net. Hold the net vertically and wait for any excess water in the net to filter out through the net's mesh. Wait until the water is equal to the point where the mesh meets the top of the horizontal stripe of the black fabric. Insert the hose at the bottom of the net into the NET bottle. Release the clip and the water sample will enter the bottle. Repeat this process twice in order to fill the bottle, but make sure you rinse the net 3 times between each toss of the net. The amount of the sample collected with each tow should be 250 milliliters, as that is the volume of the area below where the black stripe meets the net. As the sample bottle is a 500 milliliter bottle, 2 net tows should exactly fill the bottle. Place the sample into the cooler to be processed in the lab. Finally, bring your samples to your designated APCC drop-off location by 10 a.m. APCC will provide new sample bottles and data sheets for use at the next sampling event. Once home, be sure to rinse your sampling equipment well and let everything air dry. Do not let your plankton net dry in the sun because long-term sunlight exposure can damage the net. Instead, hang it in the shade, in the shed, or in the garage. Following the sampling process, APCC will inform your group of the results within about 24 hours. Thank you for all your help on this important project, and we are excited to work with you this season.
All right.
There's-
So,
Can I just
stop
kind of-
share, Jeff.
Yeah. Just kind of quickly, there were a couple of 4 different items that were in that video that might have sounded a little bit different than what I said. And one, Kevin already addressed it. BPC, because of the volume of ponds we're testing, we have our own schedule and time. So my going through those schedules and times for the teams, stick with what you've heard. 2, there will be gloves, and I didn't put it down on step 1, so that was my bad. There will be gloves in each of the kits. 3, the ice packs that'll be at the bottom will be the same ice pack. So when the 8 and 10 a.m. team will have the same ice pack, we're comfortable that it will stay frozen long enough. So when the 8 o'clock team comes back, make sure you zip up the cooler so the ice pack stays as cold as possible. And then the 4th thing is, obviously you're dropping the bottles back off at Gwen's. You're not going to any lab. So just to underscore those.
Yep. I just had 2 more things too. Roy will be in his team, of course, sampling from the boat, and their procedure will be slightly different, of course. Rather than doing a horizontal net tow from shore, doing a vertical one when he gets to the deepest point in the boat, lower the net down 3 meters and then drag it up. Same thing with the tube. The tube for the deepest point sampling is 3 meters rather than 1 meter, since you can go all the way down. And pick that up and bring it up. Of course, he'll also be having his own taking the Secchi measurements rather than the tube. The other thing is just to make sure is to make sure that the net doesn't pick up sediment or any detritus from the bottom when you're pulling the net in. You want to optimally pull it in at around 1 foot per second, but you don't want it to sink and then pick up anything at the bottom. Likewise, if you're sampling an elbow, for example, that has a lot of lily pads, you want to try to avoid, just getting a lot of plant matter in your net if possible. So also for Roy, when he's sampling at the deepest point at Walker's is only, I think, 8 feet deep, maybe at its deepest point. And
Right.
Since the net is 3 meters, you likely will only be able to lower it down about 2 meters to make sure you get a good sample that's not filled with sediment. And then same thing with the tube there. Only have to lower it down 2 meters. So Roy, if you want to communicate before you guys go out for your first shift, I'd be happy to get in touch about that.
I don't know, Roy, are you there? I'm not sure. I've spoken to Roy it's Marty again. I've spoken to Roy a number of times, and there's been good communication in his team. The other 3 members in his team are out of town, so they're unable to be with Roy. So I'll be with Roy at Upper Mill Pond and Walker's on Monday morning. So we'll take care of that, Kevin.
Yep, sounds great. And yeah, I just wanted to give you all an overview of all the ponds. We're really, it really looks good. We have some just great coverage just across the entire town. Yeah, I'm really excited for everything that we're looking to do. Karen will be helping taking some samples. Karen Malkus Benjamin helping take some additional samples at the smallest ponds, Long Pond area. We'll also be collecting an additional sample, it looks like, on the Harwich side of Long Pond. So we'll get even more coverage there. And yeah, just a lot of excellent sampling locations and a great collection of ponds that will all be in the system.
Yeah.
So yeah,
Go ahead. Sorry.
yeah, just wanted to provide a close up of those sampling locations again, if anybody wants to re-reference or look at them all.
And the locations that Kevin has here on this slide are also, as we looked at earlier, on your schedule.
Yep. Mm-hmm. Yep.
And we're pretty excited about Karen jumping in. So we're Long Pond. We could probably have 10 sites at Long Pond. It's such a large body of water, but we're excited to be testing at 2 different ponds at 2 different ends, 2 different locations at the end of the pond. So that's really good news.
Yep. And just wanted to walk through the logistics again. Monday, Tuesday, Wednesday, starting this upcoming Monday. And then the next time is 2 weeks after that. And going up until the last week of October for this sampling. And of course, if there's any sampling needed in between then for any ponds that are having blooms, we'll coordinate on that. Sampling time could be all over the place. Just put 15 minutes if you're just driving to the pond and taking the sample with your whole team, just to give you an idea of things. And yep, be sure to just keep with your schedule and making sure that all shifts are, that if you're in the first shift that you get back in time for the second shift to start, the second shift getting back in time for Marty to drive and drop up the samples.
The only thing I'd say here, Kevin, and as I discussed, the 9:45 first shift delivery time should be 9:30, as I mentioned, because the shift for 10 a.m. will be coming in roughly 9:45 to pick up the equipment. So first shift 8 a.m.ers get back by 9:30, basically.
Yep. All right. So at this point, just then, if anybody has any other kind of questions or anything else, we can look at the chat at this point too.
I have a question.
Hi, Nancy.
Hi. Is it okay to pick up, since we're the 8 a.m., to pick up Monday night and keep the ice packs in our freezer?
I'll say no, Nancy, just because I'm going to control all the ice packs and keep them frozen and have them set up in the kits, and not, if we start to have a lot of one-offs, it'll be a little difficult.
Okay. And my next question is, when you talked about scum, you know how there's that white foamy stuff that's sometimes on the edge of the pond? Do you consider that scum?
No, largely we're looking for, that might be fish foam, on the shore. We're largely looking for, you can look on our website for pictures of what looks like cyanobacteria bloom accumulations. Something that's not
Green.
stringy and filamentous like green algae, something that's not clearly, pollen and is bright yellow, but something that's, looks like green or blue paint kind of dumped on the pond, or, really just blotchy with green or blue.
Yep.
That's what we're looking for.
Oftentimes look pretty fluorescent. So Nancy, just like you saw in that video.
Yeah, I just didn't know, what is that white foamy stuff? It's not anything to worry about?
No, I think I've just heard it, as fish foam. Nothing to be concerned about with cyanobacteria.
And Nancy, quickly, I was at your pond yesterday with Liz, Carl, and Chris, and there's a lot of, as Kevin said earlier, there's lots and lots of pollen scum all over the ponds right now. So I think you'll see that right away if you haven't been down yet.
And my last question is, how do you get the GPS coordinate?
I use a compass that I have on my smartphone, a compass app that's free, and it's fantastic. And I showed that to Liz, Carl, and Chris when I was there yesterday. I can show you, in the next day or so.
I'm sure I can find it. Okay. That's
Marty,
it. Thank you.
Marty, could I ask a couple of questions?
Yes.
Good morning.
Good morning.
So my team and I are all newbies to this, and there were two things that struck me as discrepancies between your remarks and the video, and if you could clarify them. You mentioned just throw the thermometer into the water right when you get there so that it has time to read the temperature accurately. But in the video, they showed someone very carefully holding the readout portion of the thermometer and just putting the tip into the water. Is it a thermometer that can be thrown wholesale into the water?
Yes.
It is waterproof.
Okay.
Yeah, and
Okay.
we've gone through, Kevin, you may or may not know this, at BPC, we've gone through a whole host of different thermometers we've had over the years. And I purchased 3 brand new ones that are bright blue and float very nicely and can't be missed. So
Okay.
I think it's kind of fun and easy just to toss them in when you first get there and let it sit.
Okay, thank you. Second question, Marty, you talked about a turbidity measurement that we would be taking with a device that has a Secchi disc in the bottom. And I don't think that was shown in the video. So, right. So, I mean, I assume this whole thing's a little intimidating to a newbie, but I assume it will become apparent and we'll get the hang of it. But you also talked about when you do that test, wading into the water may be up to your waist. We have at our landing, we have a lovely dock that goes out to a place in the water that's certainly waist high, even a little bit more. Is it okay to do that test from just sitting on the edge of the dock if one doesn't want to wade in up to one's waist?
Yep.
Yes.
Yeah.
Yes.
Marty, I just, yeah. If
Okay.
for any sites, yeah. If there's a dock, yep, it's
Okay.
the same.
And then just let me convey the third question from some of my teammates that they put in the chat was, we have the first shift Monday morning, and some of us are early birds. How early can you get to the garage? Is 7:45 the earliest? Can one go stop by earlier?
For your first test, and just because I don't think the turbidity tube question was really answered, but it's very simple.
Mhm.
And I'll be at your pond at Seymour Monday with Charlotte and Margie.
Right.
So I'll show them, and then they're going to be able to, as we've discussed, they'll be the trainer for the rest of your team,
Right.
people that don't see it the first time.
Good.
It's very simple. It's just fill up the tube and take a look down the periscope to see if you can see the disc. But you're right, it wasn't shown in the video.
And I might just jump in and say it's something that some of our other groups don't have turbidity tubes just for price. Trying to keep things cheap for them. Sometimes they'll just take turbidity measurements by wading out to their depth and seeing if they can see their feet or something to write in the data sheet. But yeah, the method for taking the sample from taking the turbidity tube measurement is once you see it, and Marty will walk you through it. Yeah, pretty straightforward.
Good. Thank you. And then just the question of timing in the morning, is 7:45 earliest, or can an early bird show up sooner?
An early bird can show up sooner because the reason is I'll probably and expect to be at the garage Tuesday and Wednesday by 7:30. Even if I'm not there, the garage will be unlocked and open.
My one thing there is just making sure the ice packs are there. Marty, are you bringing them from your house and dropping them off?
That's a darn good point. So
Yeah.
if I know there's going to be an early bird, I'll have to be there at 7:30. So let's just say out loud now, all the ice packs will be in the kits by 7:30.
Good. Thank you both. Good.
Yep. Karen? You're muted. Sorry.
Can you hear me now?
Yep. There you go.
Thank you. If we want to watch this video again, or if we want to print out the PowerPoint because we are new at this, is there a way for us to do that?
Yeah, of course. I'll be sharing this video, the training video link. I can share a PDF version of the PowerPoint. I'll share all with Marty to share with anybody else who might be interested.
Yeah, And once this recording is finished and Kevin has a chance to download it, he'll get it to me. I will put it on the BPC website as well. So anyone can see it, including the volunteers. And as I sent around the schedule and the Zoom connectivity for this morning, I can also send the PDF of my sampling process PowerPoint around.
That'd be great.
Sure. Sure.
Bruce?
Yeah, just quickly, Marty, what time did you say the 10 a.m. team has to have the material back? I know you don't have anybody following us, but did you say 11:30?
11:30, yes.
Okay, that's it.
So the 10 team can pick up at 9:45 and should deliver by 11:30. And once you get going with this, Bruce, it just, you'll laugh at that timeline because you'll get it much quicker,
Okay.
I think.
Yes.
Margie and Charlotte?
Hi, just a quick question. So I just want to be clear, there's no waders in the kit, is that correct?
That's correct.
Okay,
Everything else will be included.
okay.
I'll go into Monday morning, so you guys don't have to wait in.
We'll go from the dock.
There you go. But the interesting thing is we like I said, I've been out to like 7 or 8 ponds, and most of them are about 71, 72 degrees right now.
Oh.
They're
Then I'll go in.
Very warm.
yeah, surprisingly warm right now.
We have a dock at Owl, but right now it's in about 8 inches of water at the far end.
Oh, yeah.
Owl is down about 3 feet from where it was last year. It's amazing.
That is true. Water levels are low everywhere. Anywhere from 1 to like 3.5 feet we've seen. Big difference.
Anybody else?
Well, if
All right.
there are no more questions, then anyone certainly on the BPC team can call me or email me anytime with questions. And as you know, I've certainly reached out to you, so feel free to contact me anytime. And if you need, the important thing with all of this is obviously the traffic and the movement and the coordination. I won't micromanage your teams, of course, but just make sure you guys have good communication, because at any given time, someone has a doctor's appointment, et cetera. Just know who's going to the garage that day, know who's going to be at the pond waiting, and et cetera, et cetera. So, and maybe that's Captain Obvious, but I thought I'd say it. And thank you all so much for, it's a great group, and everyone came to the call. I'll tell you that. Call meaning we need volunteers, and there were so many that came up. So we've got something like 45 citizen science volunteers for this program. So thanks a lot.
Yeah, thank you all.
Thank you. It was very interesting. Appreciate it. Look forward to it.
Thank you, Marty.
Yeah.
Thank you.
Thank you, Marty and